I say go for it.
Good afternoon, y'all. I'm Allison Johnson. I'm a PhD candidate at the University of Georgia, Go Dogs, advised by Dr. Brian Forschler, and I'm excited to present a little snippet of my research to you all today. There are 5 species of Reticulitermes in Georgia: R. flavipes, Virginicus, Hageni, Malatai, and Nelsoni, and members in this genus are historically notorious for displaying developmental and morphological plasticity in their immature stages, which makes identification by dichotomous key laborious and sometimes difficult. And because of the documented variability in immature stages, we adopted an integrative taxonomic approach to identify over 4,000 samples of retics to these 5 species by examining alate morphology, soldier morphology, plus CO2 mtDNA sequences verified by morphologically identified alate references. And while working on this larger project, we recognized that the soldier mandible morphology for 3 out of the 5 species of retics in Georgia consistently aligned with some simple characters based on size and shape. And so we wanted to examine the feasibility of what we felt were simple to identify morphological characters of soldier mandible pairs to reduce samples requiring additional methods of ID in any future work like this. And so we invited participants to assign over 2,000 soldier mandible pairs to one of 4 categories that had been previously identified by our ITA survey. And so pictured is an exemplary set of excise soldier mandible pairs that were scored in the survey. These are from Chattahoochee County, and you can see in each 1 millimeter square grid, there's a set of soldier mandible pairs. And this is a list of participants color-coded by experience, including 2 experienced entomologists in orange, 2 graduate students in blue, 2 undergrads in green, and 2 junior high students in gray. And these colors are going to carry through my presentation as I group participants together. And so participants were given a 1/2 hour tutorial on the recognition of the characters that we thought consistently aligned with 3 out of the 5 species of retics in Georgia. So any really large mandibles reaching about at 1 millimeter were categorized as L, which consistently aligned with R. flavipes. And then mandibles that were less than 6/10 of a millimeter, or about half of that 1 millimeter square grid box, were categorized as S, which consistently aligned with R. nelsoni. And then pairs that had a curvature to the right mandible of 7 to 15 degrees when you measure from the dorsal condyle to the tip, which was described by Lim and Forschler in 2012, were categorized as CRM, or curved right mandible, which consistently aligned with R. virginicus. And so anything else that didn't match this was placed in the not identified category. And so after training, participants were asked to complete a pre-survey test to confirm that they could recognize these characters before continuing on to complete the survey. And so each participant was asked to score a mock survey that consisted of 120 mandibles in each of the 4 categories that were randomly placed on 6 of those 4x5 grids like I showed earlier. And so the category placements from each observer had to match above 90% or at least 108 of the 120 predetermined categories in 2 attempts or less to begin the survey. So 3 participants, C, G, and H, did not achieve an above 90% score on the first attempt. And despite all 3 improving on the second attempt, participants G and H continued to have match success rates of less than 70%. And so they didn't score the rest of the survey. Once participants A through F completed the survey, category placements were scored with ITA species designations to determine matching rates among groups and individual participants. And so the large L, S, and CRM contain the number of samples called to one of those categories by participant. And then following the category column is the ITA matching rate for that character compared to the ITA determined species designation. And then the final column contains the total identified by each participant and what percentage of the survey samples were reduced from needing any additional analysis using this technique. And so nobody scored the exact same mandibles into each category, highlighting the variability of this character set. Yet the L character was the most universally recognized for all participants, followed by the CRM and then the S. And then also notably, participant C is the only one that had to retake the pre-survey test twice. and has matching rates of less than 90% for 2 categories. And so a recommendation from this experience is that all participants need to pass some sort of pre-survey test before completing, on their first attempt before completing a survey like this. And so these second set of tables follow the same format as the previous slide, but participants are grouped by experience to reduce the variability that happens among individuals. And so first shown are the experienced entomologists, then graduate students, and then undergraduate students. Below in yellow is the combined group of less experienced participants, D through F, that only took that pre-survey test once and passed it. And then below in white is all participants, A through F combined. And as you can see, combining less experienced participants decreases the number of samples identified, but it increases the number of samples that match that ITA species designation. And so we also wanted to look at the time and cost savings of using a mandible morphology approach as opposed to an ITA. And so labor was calculated at $15 an hour, and the baseline time and cost for this project was around 150 hours and $2,500 to excise, mount, and identify all samples with mandible morphology. Of course, this cost increases with participants added, but the per sample cost from this survey with all 6 participants was $1.43 per sample. Now, an ITA approach to identify all samples in this survey took around 400 hours and $15,000 to extract, sequence, and analyze all of the samples, totaling about $6.87 per sample. And so the cost savings are estimated by multiplying that per sample cost estimated for an ITA approach by identified samples that wouldn't require any further analysis using this mandible morphology approach. And so you can reduce the cost with a high degree of certainty that your species designation will match the ITA answer without the expensive cost of sequencing, depending on what kind and how many participants you have. And so experienced participants in orange would save around $7,000 and only had 2 samples that didn't match the ITA designation, as opposed to the less experienced participants, which saved around $8,100 with 19 samples that didn't match those ITA designations. So in conclusion, this quick side survey highlights that simple soldier mandible characters can be used to reduce the number of samples requiring more time and cost to ID to species. And then secondly, it demonstrates that experienced entomologists still provide the most reliable identifications, but with limited training, a small group of participants can quickly learn to recognize these characters in a survey of this style and get pretty close to an experienced entomologist with a high degree of confidence in those category placements. And I thank you so much for your time and attention. I would love to take some questions about it.
Yeah, we have a question. Do you think that you could have done this training for the identifiers virtually, or does it have to be in person?
You could probably definitely do it virtually. If you made a quick video demonstrating the training process was a set of genetically verified soldier mandibles on a page, and then we just taught everyone what the general size and shape looked like. You could definitely do it online.
And how would mandible character ID be translated into a format that PCOs or PMPs can use in the field?
It's actually it's a really practical way to use it in the field. You can just take 2 insect pens and a Petri dish in your bag and some double-sided tape. And once you get experience with excising those mandibles, it's a quick eyesight reference to identify the species, if it's either Flavipes, which was the most common, or Virginicus or Nelsonei. It's really easy to use in the field. We do it when we collect logs for the lab.
And I think you have time for one more quick question. Does colony age in starved soldiers influence the mandible characteristics? Is there anything else that you think might influence the characteristics?
I'm definitely sure it does. While we didn't look at colony age, all of these were field collected samples. I'm sure it definitely has an influence, and that's why the character set is so variable, and it's just an eyesight glance thing instead of a specific measurement to kind of account for that.
Great. Well, thank you very much. That's the questions that have been submitted so far. Raj?